Hi, everyone. I'm Kara Cameron, mom to TANGO2 warrior Bria. I am an active TANGO2 Research Foundation research committee member, as well as a regional coordinator in Canada, as well as a member of the board of directors as treasurer. Our next presenters are Dr. Vandana Gupta and Sarah Sandcooler. Dr. Gupta is a TANGO2 Research Foundation Scientific Advisory Board member and researcher at Brigham and Women's Hospital in Boston, Massachusetts. Her laboratory studies the genetics of neuromuscular diseases. Her research group has identified several new genes in neuromuscular diseases and developed animal models to test novel therapeutics and study disease pathophysiology. Sarah, a TANGO2 Research Foundation Early Detection and Diagnosis Committee member, is an MD PhD student in the pathology department at the University of Rochester School of Medicine and Dentistry. She is working on her thesis in the lab of Samuel Mackenzie, studying TANGO2 deficiency disorder in a C. elegans or a worm model, and her clinical interests include neurogenetics and the development of improved gene replacement therapeutic strategies. Welcome, ladies.
Thank you so much, Kara, and thank you, TANGO2 Foundation. We started this project a couple of years back with the funding from TANGO2 Foundation, doing drug discovery in my lab and Dr. Sam Mackenzie lab. And one of the idea was to identify small molecules that can rescue TANGO2 phenotype using different animal models, and then combine those animal models and test if those drugs could work in those different models to develop a robust discovery pipeline. So I'm going to talk about our work on zebrafish, for which we have done a small molecule screening. And Sarah will be talking about her work in C. elegans. So I'm not going to talk a lot about the background of TANGO2 deficiency disorder. Couple of points I want to make is, unlike most of the genetic disorders, the disease is not only caused by the mutation in TANGO2 gene, but it is complicated by the fact that a number of external triggers, such as infection, exercise, or some drugs, can cause metabolic crisis in some of the patients that are otherwise normal. And in spite of a lot of effort, we still do not have a therapy for this disorder, which was one of our primary motivation to do small molecule screening in TANGO2 animal models. So I'm just I like to show this diagram to exhibit the complexity of TANGO2 disease, because when we talk about a genetic disorder with a mutation in TANGO2, there are large number of genetic disorders in a single gene have been identified. And most of the genetic disorder, we see there's a mutation in a gene, and we have a very strong clinical phenotype that is not affected by a lot of different clinical or external parameters. And sometimes we also see two or more different genes, for example, in diabetes, that can cause the disease phenotype. But TANGO2 is a unique case where in many individuals, we may have a mutation in TANGO2 gene, but we may not see a very profound phenotype. But when this patient get exposed to something externally, there's a much more severe onset of the disease. And in many cases, we are not able to restore the things back to the normal level. So from the literature and the natural history studies in the patient, what we have seen is a number of external triggers, such as infection, mostly viral infections, fasting, exertion, dehydration, or the kind of diet can lead to the onset of severe TANGO2 deficiency disorder. And most of these triggers combined with a mutation in TANGO2 gene can lead to a number of different clinical spectrum, such as rhabdomyolysis, which is a muscle breakdown, neurodevelopmental phenotypes, and heart dysfunction. So we thought about doing a small molecule screen in the disease with a number of different questions. One of the things is, in patients that have been identified with TANGO2 mutations, could we prevent the disease onset when they expose to these different triggers? And when as we start to identify the drugs, can we also identify the pathways that are sorry, I just have to, can we identify some of the pathways that are targeted by these drugs, not only to improve the disease understanding, but also to identify some new triggers that may have not still been identified in many of these patients. And finally, could we discover some of the off-target effects, and as well as discover new biomarkers that can be monitored in this patient as we start to treat some of these patients in future. And I think one of the most important thing is us not only to identify drugs that can prevent the disease onset. We have so many TANGO2 patients who already have profound symptoms of the disease. Can we reverse those symptoms with any of the small molecule that we identify? So in order to do the drug discovery, there are a lot of model organisms that are available, and I'm not going to talk the advantage or disadvantage of all of them. Michael in the morning did an excellent talk talking about the model organism. But when we choose a model organism, for us, couple of questions are really critical. The most important is the disease-causing gene, human gene, is present in the animal model? And how similar is the disease-causing gene in the animal model compared to the humans? Finally, the tissue of interest is also what is the similarity between the animals and the humans? And are we able to see the similar pathological symptoms in these animal models? And depending on the different animal models, again, the choice of therapy also depends. For example, in mice, we cannot test a large number of small molecules because of the size and the feasibility. On the other hand, in small animal models like C. elegans, flies, and zebrafish, because of their small size and ease of working with them, a large number of chemicals can be tested, which we can narrow down these chemicals eventually to 1 or 2 small molecules that can be tested in mice before we take them to the humans. So my lab has been studying zebrafish as a disease model and also for developed therapies for a number of neuromuscular diseases. The advantages that zebrafish has is that the zebrafish genome is very similar to human genome, although they look very different. And whenever we breed zebrafish, we can get hundreds of embryos that can be used to do a large-scale drug screening by putting few embryos into each of these wells, each of the well of a dish. And then to each of the well, we can add one drug and see the effect of drug on the disease phenotype. And similar to humans, zebrafish have all the other organ system like heart, brain, liver, intestine. So when we talk about a disease like TANGO2 deficiency that affects multiple organs, and either by the same pathway or by different pathways in different organs, it becomes really critical for us to test in an organism that has equivalent of all the human organs, so that is easily translatable to the human patients. So keeping that in mind, few years back, we developed a zebrafish model of TANGO2 deficiency. Zebrafish have two TANGO2 transcripts, so we knocked out both of these transcripts using CRISPR-Cas9, and these are the zebrafish larvae at day 5. And what we can see is like they look really similar to each other. We have looked at number of different functions at the baseline level, and we do not see any differences between the control and the TANGO2 mutant fish, which is also seen in a number of patients that in the absence of any trigger, we do not see any metabolic crisis or any disease symptom. So what we started doing was that we started testing some of the clinical triggers that have been seen in the patient, which can lead to metabolic crisis. And one of them is exertion. So this is a zebrafish model, and the green, what we are seeing are the skeletal muscle staining. So these are the muscle actin filaments in skeletal muscle. This is a normal fish with the myofibers going from one end to the other end. And this is a mutant fish. So one of the things that was very surprising for us was that this mutant fish, they swim normally. They do not show any differences from the control zebrafish, but still they have a little bit disorganization of the skeletal muscle. And later we have seen in the data, which we have seen since published, and I'm not showing here, that they have tiny defect in the mitochondria in the skeletal muscle. But in spite of that, they live normally, they are able to swim normally. So what we did was that we induce the exertion phenotype in the zebrafish, where we make the zebrafish swim in the highly viscous water for a few hours. So after a few hours, like as expected, even the control normal fish looks exhausted and fatigued, can be seen by the lack of this nice tiration. But the mutant fish, they were much more severe when we compare to the control fish. We have seen this phenotype in two different zebrafish mutant lines now, and both of them show severe damage in the skeletal muscle on exertion. And some of the mutants, even they go to the extent that they die when we expose them to this trigger. And here is a quantification. Fish, both of the lines that swim normally before being exposed to the trigger after exposing to methyl cellulose or high viscosity water, they all crash. But the wild type kind of return to the normal level after swimming in this high viscous water. So we decided, actually, before I go to the next one, I also want to just quickly show this slide. So most of these mutant zebrafish, they die as during the juvenile phases between the similar to the childhood and the adult phase, similar to 13 to 14 years age of the human. But there are some fish that still survive to the adulthood, and they exhibit a very abnormal behavior. So on the top is a wild type fish, which is swimming from side to side of the tank, and the bottom is a mutant fish, which is just crashing. And I have a couple of other examples, which is very similar to what Sam talked in this morning about either dystonia or ectasia phenotype, where this fish, they completely lose their balance, they just crash, and eventually some of them recover and start swimming, while other they just die at this point. And this is a quantification of the swimming behavior when they are having one of those ataxia kind of episode. This is a normal zebrafish, which is swimming from side to side in a tank. And this is the mutant fish, which is crashing and just keeps swimming or circling at one end of the tank. So this is very interesting. Some of our initial studies made us very comfortable that zebrafish is a model of human TANGO2 deficiency disorder. And if we trigger the clinical stress signals like we see in the patients in this model, we can even worsen the phenotype. With that idea, we started a small molecule screening of a library of compound that has both FDA-approved and non-approved compounds of 958 small molecules. And our studies have shown, not only from our lab, a number of other labs, that mitochondria is one of the central organelle that is affected in TANGO2 deficiency disorder. Because there are a number of small molecule libraries are available, we focus on a library that is specifically targeting all the pathways that involve mitochondria in a cell. And so what we did was we took 8 mutant embryos and put them in each well of a multi-well dish. And to each of this well, we put a small molecule drug. And the couple of triggers that we decided to test in the zebrafish is fasting, which is seen in a number of patients, and exertion phenotype to identify the drug that could prevent the disease onset when we expose our zebrafish to these two triggers and return them to a normal level. So, and I'm going to show you how we do those assays. Zebrafish, when they are born, they're about a day old. They have this yolk sac that has all the nutrients they need. So we do not need to provide any external feeding to the zebrafish. But by day 5, they lose this yolk sac and we have to provide external food to the zebrafish. What we did in this case to induce a fasting, to see if the fasting would affect the zebrafish, we stopped feeding them at day 5 and no external feeding was provided. So now this fish have been in the fish water for 2 days without any food. And the second trigger that we are doing is the exertion, which I showed you in few slides back, where we put the zebrafish in a highly viscous water that has methyl cellulose for a few hours, and then let them recover for overnight. And next day we analyze the swimming behavior. And this is a summary of the small molecule screening that we have done so far. We have screened 905, we have 958 compounds, of which we have screened 350 compounds. We had some delay in the starting of this library initially because of the wrong library that was sent by the company. But anyways, we still have identified 21 drugs that are able to rescue the fasting stress, 6 drugs that are able to rescue the overexertion, and 7 drugs that work both under the fasting and the stress condition. And this is an example of the data that we are getting from our screen. What we see in the blue here is the normal zebrafish, which is the control sibling of this fish. And these are the mutant fish after fasting. So compared to the control fish, mutant fish show exhibit about 50% less swimming behavior, suggesting some kind of metabolic crisis or rhabdomyolysis in the skeletal muscle. But when we add the different drugs, some of the drugs are able to rescue the phenotype of the mutant almost a slightly higher level than the normal fish, while some of the other drugs don't seem to make a big difference. And we also identify some drugs that even are um, toxic to the mutant. So we are now making a catalog of all of the drugs, not only the one that are effective, but also the one that are toxic, because in sometimes some drugs may be prescribed to patient without knowing the long-term consequences. So it is important to know the drug that can also cause adverse events in TANGO2 patients. And on the right is an example of the same drugs and their effect on the exertion phenotype. And what we see is like the drug number 1, which is showing improvement in both exertion as well as fasting condition. But there are some drugs that are only showing significant effect in one condition versus the other. So this is also important because we talk about a lot of clinical variation in TANGO2 deficiency disorder that can not be explained by the genotype completely. So it could be that different clinical triggers or stress factor that are causing the metabolic crisis in this patient are doing so through different disease mechanism. And by doing a drug screen where we model different triggers, we may be able to come up with different drugs for this disorder and address the clinical variability in these patients. So what we have done so far, even though we have screened only a small part of the library, less than 50%, we have still put these drugs to see what kind of processes are we identifying in TANGO2 deficiency disorder. And what we see is the number of things we are identifying. For example, under the fasting condition, we have identified drugs that are stabilizing the mitochondria structure, as well as improving the efficiency of mitochondrial function. Oxidative stress is one of the consequences of mitochondrial dysfunction, and many of these drugs seem to reduce the effect of oxidative stress, making mitochondria more efficient. And finally, we also see an improvement in energy metabolism by activation of alternate energy producing pathway in the cell, or by activation of signaling pathway that control the bioenergetics. So we have this term which is called bioenergetic resilience, because when mitochondria is primarily affected in a normal cell without a TANGO2 mutation, we undergo a lot of stress. For example, if we do exercise, if we do exertion, we do fasting, but our cell have the resilience to restore the normal function, still restore the normal function of the cell, or bring the cell to a normal state within few hours. But in TANGO2 deficiency disorder, we lack this resilience. So when the body is hit by some of the stress like infection or diet, this results in the metabolic crisis. So some of the drugs that we are finding, they're improving the resilience, bioenergetic resilience of body, that even under the different stress, we do not see TANGO2 phenotypes that is normally seen. So we are still screening the library. As we are completing the screening of the library, we already have a number of compounds that completely restore the TANGO2 defect in the animal models. So at this point, we would like to test these compounds in other TANGO2 models, such as cells, animals. We are testing them in C. elegans with Sam's lab. But if any of you researchers have any models that you would like us to share this information, we'll be happy to share the information so these compounds can be tested in different models. And finally, some of the pathways that we identified are the known pathway associated with these drugs. But it may happen that in TANGO2 deficiency, they might be hitting some new pathways. And so one of our goals is to look into these treated zebrafish models either by RNA-seq or through metabolomics, and identify what pathways are getting improved when we treat the TANGO2 mutant with these drugs. So with that, I'm going to just thank all my lab members, especially Grace, who has done most of this work, Sam and Sarah, and our funding agencies, and give this to Sarah for her part of the talk.
All right. Thank you, Dr. Gupta. And I am very happy to be here talking to you about my side of this project, working on this high throughput drug screening in a C. elegans worm model. And let's see if I can drive. Yes, all right. So just to reiterate some of our thinking underlying this project, with a look at the therapeutic landscape that we know of currently for TANGO deficiency disorder. Of course, we all know that vitamin B5 has been a really important breakthrough that was identified through the natural history study, that has been so effective in reducing the number of crises for our TANGO2 warriors, and also seems to have some improvements across other types of symptoms in the disease as well. However, we also know that vitamin B5 may not be totally curative for this disease. There are other symptoms that we know can still persist even when kids are supplemented with vitamin B5. So it's really important to keep looking for other treatments that could be done simultaneously with vitamin B5 to improve symptoms even further for our TANGO2 warriors. And in addition to identifying other possible treatments for TANGO2, another advantage of a drug screening like this is in helping us to identify a possible mechanism of action for the TANGO2 protein, since the function of this largely remains unknown. And as Dr. Gupta alluded to, because we know that mitochondrial dysfunction has been identified in many studies as a potential key driver of disease pathophysiology, we wanted to focus our drug screening efforts specifically on a library that is going to target the mitochondria, which we can see a breakdown of some of the mitochondrial areas of mechanism that are being targeted by this library over here. And so this is a library of about 950 small compounds. That was also particularly exciting to us because many of these compounds are already FDA approved. So some of the ones that might come out of this study would have a high degree of a high potential for clinical translation. So to talk about the worm side of things, as some of you might have heard from us before, our lab has developed a strain of C. elegans, which are a small microscopic roundworm that have two homologs to the TANGO2 gene called HRG9 and HRG10. And so we have been working with a model that is missing both of those genes, or a double knockout model. And when these homologs are missing in the worms, they demonstrate this robust phenotype that consists of a decrease in lifespan, a decrease in reproductive capacity, a decrease in movement, both at baseline and in their ability to move in a high stress situation, such as being placed in a liquid media, and also a propensity to leave their food source, which across other strains of worms that have a metabolic function is understood in the field to be an indicator of metabolic dysfunction in these worms. For the design of our study, we also know that our double knockout worms do have an increased susceptibility to oxidative stress. So in designing what our drug screen would look at in these worms, we wanted to potentiate the phenotype of these worms by exposing them to oxidative stress using a compound called paraquat. So this is a potent inducer of redox stress, and it generates a compound called superoxide that can be very damaging to the cells. And we know that while at baseline, when exposed to a small amount of paraquat, N2 worms, or the wild type worms, are generally able to withstand this oxidative stress. Our double knockout worms are much more susceptible to this and die quite quickly when exposed to this compound. And so for a high throughput system, we are using worm death as our outcome measure. So we are exposing our worms to paraquat, seeing how many worms die when treated with paraquat alone or with one of the compounds from our mitochondrial library. We are putting them through an imager that is able to take images of our wells of worms. And we developed an automation system to count worms that are both alive and dead, which we will then use to identify compounds that are selectively improving the survival of double knockout worms when exposed to oxidative stress. And this is just a visualization of what our methods and our preparation work looks like for our mitochondrial library work. So in order to do this, we have to grow up these large populations of worms so that we can do a process called bleach synchronization to collect the worm embryos, which are grown up in liquid media and then placed on our plates containing both paraquat to induce this oxidative stress, and also our mitochondrial drug of interest, so one drug per well. And then to identify worms that have died during the window of exposure, they are stained right before imaging with this compound called Cytox blue, which stains cells and worms that have died. And so our progress through this library, we have screened through 896 well plates of compounds, which is equivalent to about 600 compounds. We need to do 12 plates total to get through the library, so we're about 2/3 of the way through. As Dr. Gupta said, there were a little bit of delay in getting things off the ground due to some acquiring some of the drugs from the company. And we also, unfortunately, lost our imaging system over the summer. But now that it's back up and running, we're hoping to be able to finish this out in the fall. And so our schematic for this is that for each strain of worms, so we have our N2 wild type and our double knockout strain, we are testing each of the drugs at 2 different doses, so a low dose and a high dose. These were selected so that we can try to get a dose that is in the effective range for, or close to being in the effective range for each of these compounds, to see if there is also a dose effect across this low dose or high dose. And then each plate is also tested with a control column that contains only paraquat. So we're able to have a control to measure the proportion of worms that die in the N2 condition and then the double knockout condition when exposed to just the paraquat. So just to give you a sense of what this looks like, this is what the data that we get from our worm imaging looks like. So here we have a bright field image of a well containing our C. elegans worms, which you can see scattered throughout here. Each of these wells contains around 100 worms. And then when we stain for worm death with our Cytox compound, you can see that the worms that have died are going to fluoresce quite brightly. And then to help with the identification of worms, living and dead. We developed an automation pipeline through a program called Cell Profiler, which is able to look at these images, and then we trained the program to identify what objects in each well are worms. And this software is able to assist us with counting the total number of worms in each well, as well as the number of worms that have died. And so from this, what we have done is for each drug, we have compared the change in survival in the wild type strain and the change in survival in the double knockout strain. And again, we are hoping to identify compounds that are selectively able to help the double knockout survive the oxidative stress while not having much change in the wild type survival. And so that's going to be the compounds that are up here on this highest point on the y-axis. And so these are some of our preliminary findings for some of the compounds that have been the most effective in promoting double knockout survival. Of course, the caveat here is that this is an N of 1. This is our first pass through this library. And so we are hoping to, once we've screened through everything, take some of our highest hitters and to increase our number of replicates to make sure that this effect is consistent across multiple replicates. And so once we finish that, we've increased our replications of our number of hits. Our next step with the worms would be to take some of our top hits and apply them to some of our other phenotypes that we can't do in as high throughput of a method. So testing if these drugs are able to rescue some of the other symptoms of TANGO2 that these worms have. All right, and with that, Dr. Gupta and I would be happy to answer any questions about our talks today.
Thank you, Vandana and Sarah. So there's a couple of questions that I think are being answered on at the same point in time. One thing that I think would be helpful to clarify is, like, based on your experience of high throughput screens, what is the typical yield going from like your complete library down to like initial validation, like stage 1, and then ultimately stage 2, like validation? What's the typical like culling of library compounds in a standard high throughput sequencing study?
So I can answer this. We have done a number of different screens in the past. We have to do a number of primary and secondary screening just to make it robust. In the primary screening, we can identify anything from 5 to 10% of the effective drugs. Having said that, in the past, we have done most of the drug screening with the libraries that can target anything, anywhere in the cell. This time, we are more focused on using a library which is targeting only mitochondria and mitochondria-related pathways that we know are affected in TANGO2 deficiency disorder. So we are seeing a slightly higher number of hits than we typically see with other screens.
And I guess that kind of goes into a second question is like, how broadly in the library that you guys have selected between reactive oxygen species and mitochondrial dysfunction, how broadly does it target things? And in your positive hits so far and your negative hits, do those pathways align very well? Like the negative hits being the ones that actually reduce survival.
So to be honest, we are still screening the library. We have not looked at the negative compounds in detail so far. We are more focused right now on the hits that we are getting with the positive compounds. And they affect very broadly, as I mentioned. They're not as broad as a completely a different library that targets everything, but these libraries, any pathway that involve mitochondria in a cell, whether cardiac, whether muscle, whether brain, this library covers a very broad range of compounds, about 1,000 different compounds that can target those pathways. Again, this was a choice we had to make because in the past, when we go with the very broad library, we did not find that many hits, but in many of the diseases, we do not have the disease information. So there's a possibility if there's some novel pathway acting in TDD, we may miss that. But from so many different labs with mitochondria as a central disease pathology that we see, I think this is a good start to go with. And we are able to see 100% rescue of the disease at this point.